Hello everyone, my name is Reed Samori and I'm here for the week 1 advanced concepts discussion in Chem 33. Happy Sunday, first week's almost over. Our topic is the leucine zipper and we have a couple prompts to address, but before we do that I thought I'd take a moment to try to explain what a leucine zipper is to the best of my understanding. So first things first, it's a common structural motif that you'd see in proteins, specifically ones maybe in gene regulation like transcription factors. They're very, very stable. They are a dimer of 2 alpha helices, so these secondary structures that we just read about, that amino acids can take on. So if this is, I tried to make it, it's not real clear, it's just a pipe cleaner, but this is supposed to be a right-handed screw. And if you take them and wind them together, you get this real stable structure. They're wound together in this left-hand screw way, like counterclockwise. And it's hard to see here, but it's actually really hard to pull apart. So even without the interactions, it feels structurally stable. So particularly with the leucine zipper, leucine repeats about every 7 so that they're always facing each other on the inside, making this hydrophobic core, which, let me see if I can find it. There it is, which is, the picture from the assignment itself, I think illustrates pretty well that leucine, continues to interact in the middle here. So you have this hydrophobic core, and then on the outside it looks like lysine and glutamate line up so that their negative and positive charges also contribute to the stability with electrostatic interactions. So just for fun, I decided to get out my molecule kit from organic chemistry. I think, yeah, there we go. I think I could show a little better what's going on and why I chose what I chose to replace leucine. So here on the right, this is leucine, and then on the left I have valine, which is my choice to replace it. And quite simply, you can just see why. Look how close these things can get to one another, so there's no real spatial problem. But more importantly, there's no charge. These are non-polar molecules. I think you have to choose something that's non-polar to maintain that hydrophobic core stability. But valine and leucine are just so similar, especially at the end. I think it just makes sense to me, even more so than isoleucine, even though it's the same number of carbons. So my choice is valine, and then I think anything that was polar would make this challenging or not stable. I just chose, looks like I chose, cysteine here, just because I ran out of molecules, and this one was an easy one to make, but it's polar, right? So you have your partially negative sulfur atom and your partially positive hydrogen here. So this would not be a happy fit. All right, and just to further bring home the point here, let's see if I can find I brought up all the lists from the book here. Here's the non-polar amino acids. I'll get on this side. The phenylalanine, tryptophan, these are just too large. The aromatics down here. And so I think we're really left with the smaller ones. And, relative to size and shape, valine just made the most sense to me. And as something that would be a terrible replacement for it, these are all the polar amino acids. Tyrosine would have been a great answer, but I didn't have enough atoms in my kit. So I just chose cysteine here. Something that could possibly replace the lysine and glutamate for the electrostatic interactions. Well, really, our choices are limited here. The only other positive one that makes sense is arginine. Histidine isn't always positive anyhow. at neutral pH. It goes back and forth. I think we learned about that in our reading this week. So I'm going to choose arginine, and then for the negatively charged acidic side, there's only one other amino acid, aspartate. So that's my choice there. And I think that covers all of the things I wanted to talk about, both prompts. Thank you for your time, and I look forward to seeing the videos of everyone else.